Hello, everybody. Jacob asked me to summarize an analysis that we did quite recently, last December, on investigating how well are genes that are potential candidates to reduce pain and suffering in mammals, how well are they conserved across other target species like cow, pig, and dog. And so what we did was to look at 14 genes, which are such candidates. And for all of them, we could identify a clear one-to-one autolog that encodes an intact protein in the other species. And of course, this is really a prerequisite to further investigate these genes. These genes tend to have conserved proteins, many of them actually really highly conserved, near identical protein coding sequences. I'll show you one example here for a gene called SLC6A2, where you can see that large stretches are identical between these 5 different mammals: dog, pig, cow, mouse, and human, and only very few isolated changes happened in evolution. The second aspect, we looked at whether these genes have close paralogs, so other genes that essentially have very similar sequences. And this is important for potential genome engineering, because then you need to take care that primers and guide RNAs are located in unique regions, that you only target gene of interest and not another gene that you want to essentially leave alone. And so there are some genes where we identified paralogs, other genes that have very similar sequences. I'll also share my screen again and show you this example here, where you can see that for this SLC6A2 gene, the other genes like A6 and A4 and A3 have stretches of sequence which are quite similar, or sometimes even near identical. This is simply something one has to take into account when one designs primers or guide RNAs. But for all of these genes, the intronic sequences and the flanking intergenic sequences are pretty much specific to that gene, meaning it is with a little bit of care, it's very much possible to target all of these genes in the genomes of mouse, cow, dog, and pig. Thank you.